Our next speaker will present the Ronald R. Tasker Young Investigator Award for the top pain abstract entitled Neuropathic Pain and Ectopic Spontaneous Action Potential Activity of Human Primary Sensory Neurons by Robert North.
Thank you. Thanks for giving me a few minutes to talk about some of my work done at Baylor College of Medicine and MD Anderson with primary afferent neurons donated from human subjects. No relevant disclosures to discuss. Really, chronic pain is often thought of as a symptom of an underlying disease. However, if you bump it together, think of it as a single disease entity, it represents a huge clinical problem, afflicting maybe 15% of the general population and leading to societal costs in the hundreds of billions of dollars, some of which estimates exceed things like cancer, heart disease, diabetes. Neuropathic pain in particular is a subset that we're all familiar with and extraordinarily difficult to treat, in large part because of poor response to traditional analgesics and modest at best response to antidepressants and anticonvulsants. Probably because of this conservative, the failure of most of these medical conservative treatments, as neurosurgeons we frequently see patients with neuropathic pain. Now, our understanding of the pathophysiology of neuropathic pain, one of the most important aspects and shared between numerous types of neuropathic pain is this spontaneous afferent or sensory activity. Really what it represents is an extreme form of hyperexcitability where neurons fire action potentials without a stimuli. And it's been demonstrated in a number of different animal models of different nerve injury, and it's often treated like a cellular level surrogate for neuropathic pain. The key question is, if we want to translate this wealth of knowledge into meaningful clinical treatments, is does this hold true for human neurons? Now, people have looked at spontaneous activity in human neurons, and it's mostly just phenomenological descriptions that, yes, it does occur. In all those previous studies, there was either limited or no clinical information about the patient's pain condition, or they were fully encompassed by patients with pain or without pain. So it leaves two unanswered questions. The relationship between spontaneous activity and the human condition of neuropathic pain, and what is the electrophysiology driving this hyperexcitability. So what we did is we used the unique access that provided from oncological spine surgery, where nerve roots are sacrificed as a necessity of surgery to obtain tissue to look at these in humans. We then paired this tissue that we collected to the clinical data for each patient and looked at whether or not for each ganglion that's collected, whether that dermatome specific to there exhibited symptoms of neuropathic pain. What that allowed is we could construct two cohorts, one of patients with pain and one of patients without pain. We can then take these tissues into the laboratory, and this is kind of a simple workflow for how I did this work. Is I take the tissue, dissect the dorsal root ganglion away from the surrounding fibrous tissue, culture the neurons, put them into a patch clamp, do whole cell patch clamp experience, and then monitor for spontaneous activity and probe the detailed electrophysiology. Now, when you combine both of these two groups together, comparing the clinical data to the electrophysiology that we had, one of the most interesting things that stands out is, yes, in fact, spontaneous activity is significantly associated with the human clinical condition of neuropathic pain. In this case, we looked at around 80 neurons from patients from ganglion donated in pain-filled dermatomes. Of those, about 15% were spontaneously active. However, when we looked at ganglion from dermatomes that had no evidence of radicular or neuropathic pain, we didn't find any with spontaneous activity. Probing further to try to decide what's the mechanisms behind this spontaneous activity driving this hyperexcitability, you look at the neurons. It takes far less a stimulus for them to fire action potentials. I like to think about this as it's like you're playing golf and you're hitting from the red tees instead of the blue tees, something I enjoy doing. In terms of once you actually get them to fire action potentials, they fire them much higher frequency for a given stimulus. So for each amount of current you pulse onto these neurons, they fire much more frequently in those that are taken from patients with pain. What's driving this is a depolarized resting membrane potential. At rest, these neurons hold closer to their trigger point to fire action potentials. So basically, in conclusion, based on all this data is, yes, in fact, this is a cellular marker for human neuropathic pain, and really it opens a gateway for these methods to be used to, for instance, screen different novel analgesics in human tissues prior to going into things like expensive clinical trials. and that this hyperexcitability is driven by, one, the action potentials need a decreased stimulus to fire. They fire at increasing frequency in patients with painful conditions because of these depolarized resting potentials and a hyperpolarized threshold. These are the acknowledgments from the group in the pain medicine at MD Anderson, and of course our neurosurgical teams that helped to donate the tissue. Thank you.